Welcome back to the pharmaphorum podcast, or indeed, welcome if you're tuning in for the first time. In this episode, I speak with Rafael R Carazo Salas, founder and CEO of CellVoyant, an AI-first biotechnology company looking to ameliorate the stem cell industry landscape and get these life-changing treatments to patients. From the essential biology of stem cell types to the wide-ranging potential applicability of this field of research when it comes to a multitude of disease areas, Salas is certain that stem cell therapy could help heal the world. But with such hope come many very real and very complicated barriers. Listen on to learn the ins and outs of the conversation. By the end, I'm sure you'll agree that the sky might very well be the limit, but we've got to gather momentum and some altitude yet to fly there. As ever, thank you for listening. This is web editor Nicole Raleigh, and today I have with me Rafael R Carazo Salas, founder and CEO of CellVoyant, an AI-first biotechnology company spun out from the University of Bristol. CellVoyant's mission is to help power the creation of novel stem cell-based therapies by making their development cheaper, more effective, and more accurate. Today, Rafael and I will be discussing the stem cell therapy industry and the importance of making treatments more accessible and affordable, particularly when it comes to currently untreatable conditions. But before we begin, Rafael, perhaps you could tell listeners a little bit more about yourself, about your journey to what you do today with CellVoyant, and I believe you originally wanted to work in the film industry.
Yes. Well, first of all, thank you very much, Nicole, and it's great to be able to speak with you and with all listeners. It's a truly pleasure to be here. Yes, that's right. Absolutely. So I'm an interdisciplinary scientist. I trained in mathematics and physics and biology. I have more than 25 years of interdisciplinary experience training globally. And it was a convergence of many of the areas that I worked in particular in more recent years in stem cell science and machine learning that pushed me to found CellVoyant in 2021. And that kind of made what CellVoyant does possible today, those breakthroughs. But actually, my story was not rectilinear. It didn't go directly there. So, I'm born and bred in Costa Rica. I'm naturalized British now since nearly 20 years. And initially, actually, I didn't want to do science. I wanted to do arts. I'd been doing amateur theater at school for 9 years. And I was very much into performing arts, and I loved particularly the visual medium. And so, in fact, my plan was to study cinema and theater abroad in Paris and New York, but I didn't get any scholarships for that. But I happened to be good at maths and science, and so I did get scholarships for that. And that's what made my career pivot, basically. But that passion for films never actually left me. And in fact, with biology, what really ignited my passion for the first time is when I saw cells under the microscope grow, move, and divide, and so on. And, fast forward 25 years, the machine learning models that we develop at CellVoyant are trained by filming millions of cells that are growing and dividing and differentiating under the microscope. So it's just different types of movies, but that stayed with me all along.
Gosh, yes. So the visual medium indeed was your passion all the way through, I can see. Thank you, Rafael. Right. So if we turn now to stem cell therapy, for those who aren't aware, stem cells are types of cells that have the unique ability to self-renew and differentiate into more than one cell lineage. Stem cells can be extracted from different sources, including bone marrow, amniotic cells, adipose tissue, umbilical cord, and placental tissue. They are considered the building blocks of tissues and organs, and over recent decades, since the 1950s, they have been studied and utilized for biological repair and in regenerative medicine, including epigenetics and dentistry. However, Rafael, perhaps you could go into the finer details of the types of stem cells, the totipotent and pluripotent, multipotent, oligopotent, even unipotent, and of course, their medical usage potential.
Absolutely. Thank you. So let's start with the basics. So a stem cell is just a type of cell that is not specialized and that actively proliferates by cell division to make more and more stem cells, more of themselves. Every time you and I get a paper cut in the skin and cells are destroyed, then they will have to be replaced by new skin cells, let's say, and the production of those cells is possible thanks to our body having stem cells. which in that situation help to continually replace and replenish damaged or dead skin cells, in that case, in our body. Now, stem cells are there with us since the very first moment we're conceived. So at conception, the mother's and the father's reproductive cells fuse, and then they give rise to one very special type of stem cell that we call totipotent cell, a cell that has the potential to engender everything, all cell types in our body. And so from that cell, actually the entire body, your entire body, my entire human body will eventually emerge. So that totipotent cell is at the very start. And then, one cell becomes 2, that become 4, 8, 16, 32, 64, 128, and so on and so forth. And by the time a big mass of cells starts to form, then we start to have bigger features emerge, like a front and a back, a top and a bottom, a left and a right. And coincident with that, then cells start to become more specialized, regionalized, and specialized in the function. And stem cells themselves, they take a more restricted potency. So if totipotency was the capacity of a stem cell to give rise to all cells and tissues that make the body, next up, let's say it's pluripotency. We find, for example, in the early blastocyst embryo, these pluripotent cells will give rise to most cell types in our body, just not extra embryonic cell types and tissues. Then comes multipotency, which is again more restricted. An example of that is what we call hematopoietic stem cells that can give rise to the blood lineage, for example. And then comes oligopotency. Now, this just means few cell types can be derived from those stem cells. And examples, for example, lymphoid cells that can give rise to B and T cells, but not red blood cells, compared to the hematopoietic stem cells, which can give rise to a much broader lineage. And then all the way at the extreme end of that is what we call unipotent cells that you were mentioning. And these are cells that essentially have the capacity to differentiate to basically just one cell type, like hepatoblasts going into hepatocytes in the liver. And so, and if you make, if you think about it, it makes sense that as we develop, as we grow, the stem cells that are found in particular in adults should be restricted in the potential, because if I make a paper cut in my arm, it would be tragic if I get a piece of eye in there. So actually, it makes functional sense that those stem cells should be restricted. But then, of course, it means that for therapeutic purposes, then different types of stem cells have different limitations. Now, every disease essentially boils down to a dysfunction of our cells and tissues in our organs. And so you think it's natural that what we would like to do is basically to fix cells, maybe with other cells. Now, we're used to being treated by drugs to alleviate or cure ailments. I take an aspirin because I have a headache, and every molecule of aspirin is identical to every other molecule of aspirin. And aspirin cannot make more aspirin in our body. They're simple, they're targeted. But actually cells, if you think about it, are much more complex. A cell can make more cells. A cell can migrate to a place where it needs to go. And so they should be the ideal drug in a way. And that's what cell therapy is about. It's about using cells to cure, cells as drugs. We don't think of it actively so much. We're used to thinking of drugs, but actually we are used to cell therapy in the form of, blood transfusion. It's a very, very bulk type of cell therapy to alleviate, let's say, low red blood cell count. Bone marrow transplant, that's been around for 100 years or so. In its modern version, the idea is that by harnessing things like stem cell technologies, pluripotent stem cells, multipotent stem cells, we may be able to derive from those cells functional specialized cells that can then be used to alleviate, substitute, supplement, repair damaged cells in our bodies and tissues.
Thank you for that. Gosh, yes, definitely don't want an eye when you have a paper cut. Yes. No, but this concept of cells as drugs is quite a powerful one if we do specifically put it into the words there. So now let's think about the European Medicines Agency, the EMA, and the US Food and Drug Administration, or the FDA, have both set good manufacturing practice, or GMP, guidelines for safe and appropriate stem cell transplantation. And stem cell therapies are at present indicated for a range of clinical conditions to treat, for example, you were talking about blood earlier, genetic blood diseases. And another emerging use for stem cells is their perceived potential to treat pain states and even neurodegenerative diseases such as Parkinson's and Alzheimer's disease. Stem cells offer then, I think it's fair to be said, hope, in short, for currently untreatable conditions. So perhaps we could go further into those, perhaps the latter conditions I mentioned there and developments that are coming to the fore.
Absolutely. So, cell therapy will help heal the world if only we allow it to, because because we can do so many things with cells that we couldn't do with drugs. A prime example of a cell therapy is a cell therapy against blood cancers called CAR T cells, which have really revolutionized the way we can treat certain types of blood cancers, acute lymphoid leukemia, and others. But of course, cells and stem cells can be used for many other applications. So you mentioned, for example, neurodegenerative diseases like PD, like Parkinson's disease. Basically, the idea is that the loss of a certain type of neuron that is called a dopaminergic neuron, which is important to regulate dopamine, which was important for many body functions, including movement and thinking. The idea is that we could use stem cell therapy to replace those failing dopaminergic neurons that are found in the body that are degenerating, to then substitute for this function that's appearing. And as I said, there are many technologies these days that can be used to derive in the lab functional cell types, including many different types of neurons in vitro. And if we could only apply them to humans, then they could restore our dysfunction. There is no, in the case of Parkinson's disease, for example, it's very promising. There's no approved stem cell therapy for PD, but it is highly promising. And of course, imagine how disruptive this would be. I lost my dad to neurodegeneration actually recently, and it is very frustrating because you can make so many cell types in the lab, you just cannot use them as treatment. Imagine, for example, spinal cord injury. That's another thing, right? Where if only we could produce the types of nerve cells that could restore the disconnection that there is during spinal cord injury, then it would be again disruptive. It would be again a watershed moment. But beyond neurodegeneration, there are many other areas where there's a lot of progress. For example, there are ongoing clinical trials with therapies for type 1 diabetes, where for example, it's been possible to produce pancreatic types of cells, pancreatic islet beta cells in particular, that will secrete insulin, and that allow to the patient in the clinical trial setting to not have to inject themselves with insulin every day. I mean, one of the early clinical trials, again, a watershed moment, demonstrated the 65-year-old male patient who, from one day to the next, was cured of this until then incurable disease. So there's a lot of promise. There's also a lot of responsibility to do things right, because, of course, we were mentioning GMP, good manufacturing practice, and of course, it's important that as these therapies are generated, they are, of course, of high quality, but also appropriate for their use and for clinical trial use. They need to be safe, right? Purity, potency, safety, much more so than drugs in a way with cells. The unintended consequences need to be really thought through and really tested for. So lots of promise, but lots of caution as well. Any failure, any, let's say, instinct to rush and get a therapy that may have unintended consequences could set back the field by a decade. Like for drug discovery, the development of cell therapies takes typically 10 to 15 years to go from bench to bedside. And so you can imagine that being set back by 10 years as a field would be extremely damaging. So lots of promise. The work needs, of course, caution in particular, because these are live materials. But of course, the result, if done properly, could be revolutionary. I mean, think of Casgevy, that was approved in this country as a world first in November, I think it was, for sickle cell anemia and beta thalassemia. Very expensive. We can come to that later, but life changing, right? It could restore a function that was completely absent. Things that were unthinkable even a few years ago. Yep.
Absolutely, and thank you for sharing your own personal experience with all this.
Thank you.
So, if we focus now on the notion of progress and turn specifically to CellVoyant, the company secured £7.7 million in seed funding last month to continue advancing its technology, which includes artificial intelligence, or AI. So, Rafael, firstly, what's the experience of, let's go back to the roots of CellVoyant. Let's discuss what's the experience of being a university spin-out like? How does that model work here in the UK?
Yes. So first of all, let's start from the beginning. The UK is an academic powerhouse. One of the things that it's globally recognized for is its academic institutions. Lots of extremely high-level training, and with that, extremely high-level innovation and invention are happening. And so, as such, actually, the UK is uniquely placed to spin out companies. Now, when it comes to the practice of spinning out companies, then of course there's the practicalities of it differ from country to country. Everybody cites Stanford and the US in particular as being a particularly conducive environment. I think the UK is a good environment in that there are mechanisms to spin out companies. I can tell you a little bit more about my specific experience with CellVoyant, but there are ways to sort of protect intellectual property, and there are mechanisms within universities, and there are funding routes to do this, including support from the government. One big difference between the UK and the US, for example, is the attitude towards equity that universities, in this case, aspire to get from the foundation of a spin out versus their counterparts in the US. It's a very contentious discussion, but one that I think will lead to very healthy changes. So traditionally, there's been the mechanisms in place have tended to charge, so to speak, very high equity for founding a company, but on behalf of the institutions, in a way that leaves very little for the founder already to start with, even before they actually raise funds. Famously, there was this number, I don't think it's true anymore, but of Oxford University taking 55% equity even before money. And of course, that's not really an incentive for founders to found companies. This has changed, and it is changing a lot, including at the University of Bristol, where I am, where now, universities are realizing, like in the case of the United States, that it's better to have 5% of a billion than to have 50% of 10,000, right? So you need to encourage and give mechanisms that are permissive and inspiring for founders to found companies. Now, in the case specifically of the University of Bristol and Bristol as an ecosystem, Bristol has got a lot of things going for it. Universities are thriving, has got a thriving enterprise ecosystem, unlike, the Triangle, Oxbridge, and London. So I spent a long time in my career in Cambridge, where I had my group, in fact, before moving to Bristol for many years. So I know firsthand the experience there. It's very, very dense. Those environments like Cambridge and Oxford and London are very dense. But as a consequence, there's very hard to get space, for example, laboratory space to house companies and so on. In the specific case of Bristol, it punches above its weight. There's lots of incubators being built in town to give oxygen and location to thriving new companies. And as I said, the university itself, the University of Bristol I'm affiliated to, is investing heavily in enterprise, moving towards enterprise and helping catalyze this transition from academic to translation of innovation. So yes, and then there come other considerations that are beyond the institutions, and I think these are very important as well, that have to do with, for example, government. It's really interesting how some things can have completely unintended effects. For example, recently there was this planned change to salary thresholds for skilled worker visa, and as a way to tackle immigration, so on and so forth. In the case of academia, a lot of the workforce that we get in academia comes from postdoctoral researchers that come from abroad. And the salaries in academia are not particularly high, in particular in the UK. And so these kinds of measures actually could be made better to make it easier for people to come out, because although we are, an academic and an innovation powerhouse, we do need talent coming from all over the world, the best talent really, to be able to thrive and to make that promise a reality. And that goes beyond the academic institutions, requires support from the government and legislation that is conducive to talent being attracted here as opposed to elsewhere. Yep.
Absolutely. So if we think about talent, we think about the value of that talent. Let's now switch our conversation to sort of costs, or at least the finances. And that goes sort of hand in hand with what AI can do to a certain extent, because AI is currently providing significant benefits in the global healthcare ecosystem in numerous ways. For example, determining viability, functionality, biosafety, and bioefficacy when it comes to our topic today, stem cells, as well as appropriate patient selection. And applying AI permits precision and accuracy, which goes back to your trio of words earlier of purity, potency, and safety. So on the application of AI in stem cell therapy and a focus on affordability, coming away obviously from the academics themselves, can you tell listeners how it is believed this technology will be able to affect cost saving up to between $10 and $100 million per therapy? And for listeners, that's about £8 to £80 million, if you were wondering.
Yes, so cell therapy is out to heal the world, if only we allow it to. What do I mean by that? Well, let's say Casgevy, again, this sickle cell anemia and beta thalassemia therapy that was approved in the UK in November of last year, that is projected to cost about $2 million a dose. In the case of that specific therapy, one dose should suffice. But if we take other cell-based therapies like CAR T therapies, again, they cost each dose, let's say $500,000. So it's very, very expensive therapies. So who's going to be able to afford them? Obviously not individuals, maybe the NHS, if the therapy is approved by the NHS. In the case of countries like the United States, insurance agencies may or not be able to reimburse this. The reason why these therapies are so costly is superficially not dissimilar to drug discovery and why drugs end up being so expensive, to go from an initial drug all the way to an approved drug. To make a new drug, it is said that it can cost $1 to $2 billion. And that's because at every step of the way, there are many assets, many drug leads that you don't know which ones are going to be the winners at the end of that race, if you see what I mean. And so lots of expenses in testing and de-risking take place through, a decade or more until you get to this one drug. And then the drug companies will need to recoup that cost somehow. This exactly the same thing superficially is going to happen and is happening already with cell therapies. Not for the same reason as drugs, but because cells are so complex, being able to control, ultimately you want a dose of curative cells put into a person. And so you need to have the right number of cells with the right type of functionality, high enough purity, and low to none unintended consequences. These things are very, very hard to control with cells. Because cells are so expensive to produce, if again, it's a race where only a few of those cells that are being developed into a functionalized cell type that could be a medicine, only a few will make it at the end. Being able to understand which of those you should remove from the race would reduce the costs enormously. Why? Because maybe some of those cell lines that you were pursuing in parallel don't proliferate very well, and you can't really tell at the beginning, but eventually they die out, or maybe they don't differentiate into specialized functional cell types well, and you cannot really tell at the beginning. It's very black boxy, the way we develop and manufacture as an industry these cell therapies. And this is where AI comes in. So, CellVoyant is an artificial intelligence first, AI first biotech company. Our mission is to accelerate the development of novel cell therapies with the help of artificial intelligence. And it's what we're doing, why we think will help save tens to hundreds of millions of dollars per therapy, is because we are developing technology that allows us to open that black box and see in real time by using computer vision, so automated image analysis, together with artificial intelligence that learns patterns from those images, from those movies of cells, that allows us to see in real time, as we are deriving these functionalized cell types, why so few are becoming the type that we want, why the yield is so low, why it's taking so long, maybe which cell lines are the right ones to use for a given application. As I said, we have hardly any predictive power, any way to see ahead of time, which we should pursue that are going to make it all the way to the end. CellVoyant is developing predictive artificial intelligence-based technologies that will allow us to select better cells at the start of a cell therapy development and manufacturing processes, improve and optimize the derivation of the target cell types to improve yield, so increase the cell number at the end, reduce the time of derivation, and therefore reduce the overall cost of derivation. The ultimate goal of that will be, specifically, as you mentioned, tens to hundreds of millions of dollars that are currently invested at key points of those 10 years development pipeline of cell therapy are being wasted because we don't know how to choose the right cells at the start of the pipeline, because we don't know how to optimally differentiate the cells into the target cell types. If we can make a dent at any and all points of that process, then we can make cheaper therapies that take less to make, that will eventually reach out to more people to treat more conditions. That's essentially our goal, and that's what we think AI will allow us to do.
Thank you for that. And I duly note that you went to the patterning within movies there. So that film passion is still with you, Rafael.
Yes,
but
that is.
To close, let's focus on that keyword perhaps of acceleration and going into the future. What of the future? With all this sort of speeding up, this velocity, what's next for stem cell therapy as a broader remit as that encompasses? Is the sky veritably the limit?
Yes, in many ways it is. I mean, cell therapy, as I said from the beginning, is out to heal the world, and in particular technologies like pluripotent stem cell technologies, and one particular type that we call induced pluripotent stem cells, iPSCs, that were discovered in the lab in 2006. They won a Nobel Prize in 2012, the discoverers of this technology. Clinical trials were started with this technology in 2014. That is a technology that allows us to make stem cells from you and from me that are pluripotent. The adult stem cells tend to be very restricted in their potency, as we discussed potency at the beginning. These pluripotent stem cell technologies allow us to make from you and from me stem cells that are almost like from the embryo. And therefore, we could derive retina, we could derive kidney tissue, we could derive pancreas, heart, liver, neurons, all from your own cells one day. So the sky is the limit in that the promise of cell therapies, in particular, powered by these new stem cell technologies like pluripotent stem cells, should allow us to generate functional cell types to treat all sorts of diseases beyond cancers, cardiovascular diseases, infertility, neurological diseases, as well as neurodegenerative diseases. So I mean neurological diseases like spinal cord injury, versus neurodegenerative diseases like Parkinson's and Alzheimer's and so on. And of course, metabolic disorders like the one I mentioned, like diabetes, for example, type 1 diabetes, where there are many assets that are progressing very promisingly. Age-related diseases like age-related macular degeneration, loss of eyesight with age, where in particular for the pluripotent stem cell powered cell therapies, these are very, very mature. The first clinical trial using this technology, these induced pluripotent stem cells, was in 2014 to target age-related macular degeneration, to make retinal cells to replace damaged retinal cells. That's very, very mature and coming online soon. And of course, cancers. So there's so many areas where we can literally transform health and disease. Aging, of course, is another angle. If we could replenish and substitute in our body age cells with new cells. So the sky is the limit, but only if we allow it to. So if only we can find ways to develop and manufacture more efficiently in less time with less cost these cell therapies, then we could literally alleviate and heal the world in all sorts of new ways. So the future is bright, but we need to help the future. And this is what we're out to do with CellVoyant.
Thank you, Rafael. It's been a pleasure.
Thank you so much. Thank you very much, Nicole. It's been a pleasure for me. Thank you, everybody.
And so that concludes another episode of the pharmaphorum podcast. You can find out more information about this episode, including a download link and information about previous installments of the series at pharmaphorum.com/podcasts. The pharmaphorum podcast is also available on iTunes, Spotify, Acast, Stitcher, and Podbean, where you can find and subscribe by searching for pharmaphorum. Of course, don't forget to visit our website itself, where you can sign up for daily news and analysis bulletins, and follow us on Twitter, or X nowadays, at pharmaphorum. That's all for now. Thank you for listening.